So today I'm going to be showing you a little demo on how to annotate the results from our sequencing experiment. So first I wanted to just point out a few resources. There's an overview of the lab, the online activity here. There's a week 2 protocol which details some of the information that we're going to be talking about. And then there'll be some information on background, and make sure you watch this lecture on the genomics lab first. I think it'll help provide you a lot of context to what you'll be doing. The data is located here. There are 2 data files. There's a sequence lengths file and a file that has the actual sequencing results, some of the sequencing results. You're going to use this sequence lengths file for this R cloud exercise, and there's a tutorial here that you can link to that will show you how to create an R cloud account, which you can link to R cloud through this link. And then there's instructions on how to make an actual histogram and so generate some other summary statistics using RStudio. And there's the code that you would actually use here as well. So feel free to watch that video and then use this to create the figure and generate the statistics that you'll need eventually for your paper. Additionally, I'm going to walk you through how to do this genome annotation component and specifically how to identify a sequence to use, how to blast the sequence, and how to use DFAM to look for repetitive elements in the sequence. And then lastly, I'll highlight the table that where you all need to make for your papers about the sequence, about your annotation information. So let's get started. First, I'm going to download these sequences. So if I go to this link, if you're logged in as a CSUMB person, you should be able to download this link. And this should be the file. I'm just going to download this file. And I'm just going to put it on my desktop for now. So this file that I'm downloading is a FASTA sequencing file. It's all sequences that are over 20,000 base pairs long. So if I open up this file, there's a couple different ways you can view it. I'm going to open it up with any text editor. I'm going to use a program called BBEdit. If you're on a Windows machine, you could use Notepad. If you're on a Mac, you could use TextEdit or Text Wrangler or BBEdit. And I'm going to open it this way. You may also be able to open it here online using the Google Text Editor tool, but it may not work for everybody, but it's another option. When you open up this file, this is what it looks like. This is what DNA sequencing is, how DNA sequence data is often stored. It's in a format we called FASTA, and in FASTA we have a identifier here, right? And this is a sequence ID, a unique identifier for every sequence. This is an important number for you. So you're going to note this number, and you're going to have to identify a unique sequence, 3 unique sequences for you all to look at. So this is the unique identifier, and then we have our genome sequence here. And as we go down, we'll have an entire sequence, and we'll go until we hit the next little less than sign or this little caret here, right? And then we're going to have the next identifier for the next sequence, and then the following sequence after that. So you'll have to go through and identify different sequences to use, and so it's really important, once you identify a sequence that you want to use, that we're going to go back to the web page. And I can't stress how important this is, that we all not do the same sequences, otherwise you're going to have to repeat work. And that you go to this sequence sign-up sheet. When you go to this sequence sign-up sheet, you'll be able to take your sequence and paste in the identifier, right? And then you'll put your first name, your last name, and the section that you're in. And then if you have any issues with your sequence, you can put a little notation there so we can follow up on it later. But this is just for if something comes up. Of course, everyone's going to be looking at the same sequences, so it's really important that once you put something in, that you do a quick search. And if I find something, I'm going to use the find and replace option here that you can find under the edit menu. If I hit find, and so I've got it in here, right? And if I hit find, I'm going to find it, and I find it, right? I found this one result. So it's already in there, right? So I found it once. That means that I have to go, and I should go, instead of trying to continue on with the same sequence, right? I'm going to have to pull up a new sequence. I'm going to have to look for something new. So, you know, I'll have to go down. Maybe I'll take the next one, or I'll just scroll down to somewhere else again, but I'll find this, right? And when you're doing this, please ignore don't include this little caret. It's going to complicate whether or not you can search things appropriately or not. Remember, it's everything after this little caret. And so here I can go, again, I can find, I can look, and see it says there's no entries that for matching, right? So then it would be safe. Then I could go ahead and put this in and use this as my new sequence, right? And so I'd have to go do and identify 3 sequences. But for our intents and purposes, I'm going to use that first sequence as my sequence to start with. So I'm going to go through, I'm going to copy this entire sequence that goes on, it's on these 2 lines, line 3 and 4. So I'm going to copy these sequences. And then I'm going to go back here, and I'm going to go to the page, and I'm going to click on this NCBI BLAST web page. This is going to then take me to the BLAST, the basic local alignment search tool, which is sort of the most basic way of doing bioinformatics searching in sequence analysis. So here I can use my query, my nucleotide unknown sequence, and look at all the other sequences that are out there and see if it's similar to any of those things. There's a couple different ways you can run BLAST. We're interested in identifying coding sequence, protein coding sequence in the genome. So I'm going to use this BLAST-X tool, which is going to take my nucleotides and compare it to existing protein references. So what I'm going to do is I'm going to go over here, I'm going to paste my sequence into this query section. So this is the thing that's unknown, right? And I'm going to compare it to a group of known sequences. There's a bunch of different options here. You should select reference proteins, right? So these are the proteins that are in RefSeq, which is a very well curated database for protein sequences. So I'm going to put it into RefSeq. I'm also going to go down to these algorithmic parameters, and I'm going to select the max number of targets. I don't need 50, 100, 250 targets. It's going to take too long. So I'm going to select 10 targets, and I'm going to set this expected threshold, which says how likely, this is the threshold that gives me the probability that any match that I found was just found at random. So here I'm going to give it 1 times 1 e to the minus 0.5. So another way of writing that is 0.1, 2, 3, 4, 5, right? That would be the same level of threshold, 1 to the 10 to the minus 5. And so right, this is going to be the threshold that I'm going to be using. This will allow me to have a very low probability, 1 to the 10 minus 5, that whatever match I found was found at random. So once I have these things listed, I'm then going to hit this BLAST button. Once I do, it's going to start searching the database for matches of my coding sequence to protein sequence. So it's going to be translating my DNA sequence and looking across it for any sort of sequence that could be related to protein coding data. And in this way, we can help to identify where we might be finding exons or coding sequence in our genome, right? Because remember, we're looking at genomic DNA, so we should have a code for genes in there, but genes in eukaryotes aren't listed just sort of as one continuous segment. They're broken up into exons with introns intervening between them. So we'll see if we can find any evidence of protein coding sequence in our genetic sequence, because we don't know what genes to expect and what kind of protein coding information we might expect to find there. In addition to doing this BLAST search, we're also going to look for other genetic elements in our DNA sequence. And so this is for this, we're going to use this DFAM tool. While BLAST is helping us identify possible protein coding sequence, DFAM is going to look and see if we can find any evidence of repetitive elements, things like those PV92 elements that we amplified in our cells, right? And so this is going to help us find repetitive elements, which are also important in our understanding of genetics and genomics. And what we're going to do is we're going to paste our sequence here in this sequence box. And then DFAM is going to use a bunch of different tools, but it's largely based around a search algorithm called a hidden Markov model approach. Not that you need to know that, but basically that you know that it's going to look for good matches. We're going to take a query organism here, and we're going to pick Danio because this is another fish species. This is zebrafish. All these other organisms, there are a bunch of other organisms here. Danio is one of the most well-known and well-researched fishes, so it'll be a good one for us to choose as a comparison species. There are a wide range of other taxonomic groups here, but Danio is probably our best when it comes to getting a good genomic comparison. We're going to stick with this DFAM curated threshold. This is a threshold that, like an e-value, is basically where DFAM says if it's going to give us an estimate of what's a legitimate hit, what is a good, reliable result. So I'm going to hit submit here and allow this to continue. So this job will now be submitted to DFAM, and it's going to do some searches and try to identify these repetitive elements. All of these methods are basically using sequence similarity in order to identify these elements, right? They're looking and saying, okay, you have this sequence that's unknown. What else out there is similar to it? What else out there is like if I'm looking at coding sequence, what are the other examples of coding sequence that are similar to this one? What are the other examples of repetitive elements that are similar to this one? And it's using that level of similarity to say, yes, these things I think are the same or they're not the same. And that e-value that helps establish that threshold of what is sufficient amount of similarity for these things to be identifiable. If we look at the results now, we can see, for instance, this example, we didn't find any significant results. And that's fine. That might happen when we look at anything. We might not get a result. And this would indicate that there's no evidence of protein coding sequence in this sequence. It's not, it could definitely happen, right? So that's fine. That means that we don't have to put anything in there. I'll put another sequence in here so that we can look and see if I can find, show you an example of when we might actually get a result as well. Just give me 1 second. One of the things that we note is that when we look for repetitive elements, so we didn't find any coding sequence in this gene, right? Which means that there's no evidence that there's actually a gene located on it. That might not be surprising. You know, in humans, only about 3% of our genome is considered to be coding sequence. So having only, you know, a small, having 20,000 base pairs where we don't find any evidence of coding sequence may not be all that surprising. After running our search in DFAM, we can see that some results, some matches are reported. So some regions of this sequence have matched to known repetitive elements, and we can see a table of these results here. These are the matches on our sequence of our unknown sequence to known sequences. As we go through and look at the results, we can see that there's a bunch of information that's reported, right? We have some information identifying what family it belongs to. You can click on this, and one thing that you'll want to note is you'll want to save this information. We'll need to know what the identifier is, and this is where you can find that identifier. Additionally, we'll have this information on what this element might be. So it's P-1N3DR. This is a DNA transposon from zebrafish. And we can see that there's a bit score associated with it, which is sort of a way of scoring how well of a match we got. That same sort of e-value information, which tells us that, you know, anything below, this is the probability that what we got is just a match at random. And then we have specific alignment positions. One thing that you can do is you can sort by these alignment starts, and you'll see that as we go along, you'll see each of these different elements. So for instance, this P1N3DR, right, this is this element here. Note that it's on the bottom of the strand, and because of that, the alignment end is actually sort of before the alignment start. The coordinates on this sequence start at 0 or 1 and go to the end of the sequence. But DNA has two strands. It has a positive strand on top and a negative strand on the bottom, right? So it goes 5' to 3' here and 5' to 3' on the bottom, or in the opposite direction, as DNA is antipolar. And this element here is on the negative strand, and that's the reason why the alignment end is on this end and the alignment start is on this end. So it's important to note that information as we go through and identify this. So what you're going to do is you're going to take these results and the results from any blast results and put them onto a table that you'll be analyzing and using for your paper. You can ignore the simple random repeat information on this page, by the way. It's not necessary to look at. But it is indicated here in these black regions. If we looked at another element that I had blasted, we can see that there is some blast results for this example. What we see is there is another rockfish. There's data on another rockfish that indicates there is some protein coding information. We can click on the graphic summary and it can show us where on that sequence we might see that. And so here what we can see is that there's this one region that has a hit to protein coding sequence. We can look at those specific. They are ranked in the order in which they are likely to be matches. And we can see that this alignment score tells us how good of a match it is. So this is a pretty good match that we found. If we click, we can get to the alignment either by clicking here, it takes us to it directly, or we can click on alignments and it'll show us the alignments as they are ranked in order. And what we can see here is we can see where the position of where the alignment is. So one thing that we could notice if we look at this directly, right, we actually have for the first hit for this RAS association domain containing protein 5 isoform 3.x3, right, we see that there are 3 actual ranges that hit, right? So we can see that there's a section where we see a match to the protein, to this RAS associated protein from Sebastes umbrosus, which starts at amino acid 109 and goes to amino acid 219. And we see there's an also match that goes to amino acid 89 to amino acid 110. And we also see a match where it goes from amino acid 218 to 238. Right, so there are some parts obviously that are missing, but what this does tell us is that when we look at the sequence, notice that the sequence, again, it's going to be on the negative strand, so it's on the bottom strand of this sequence. This is the start position and this is the stop position, right? So it's the query finds us, it has a higher number here and a lower number here because it's on the negative strand, but we can see that this is the start and this is the stop. We have another start and stop, another start and stop. There is some evidence that there are some overlap regions where these are starting and stopping, and this may be due to some insertion deletion mutations that we see in our sample. But overall, this is promising as we found some good results to what we were looking for. We can look at this alignment. It shows us everywhere there are matches and everywhere there are mismatches, places where we have similarities, where we have 2 amino acids match up that have similar chemical characteristics. And it also shows us regions that could be missing from the sequence. So for instance, this looks to be there's a large deletion that's absent in this that's present in this organism but absent in the other. So Looks like we have a good match. Now we got to take all this information and put it into a table. And the table that you guys are going to build is going to be something that's going to look like this. This is an example of that table. If we look at the sample of the table, this is from a previous example, right? This is information that you're going to need for next week. So you really need to make sure that you have this already done before you come to lab for next week, because this is the information that you'll have to include. So if we take our sequence, we have our first sequence, our query sequence, right? We're going to identify the database that we found. And what we've done for the first sequence in this case is we've listed all the elements that were found on it, organized by the order in which they appeared. Notice a couple things here. So we have identifying which database, whether it's DFAM or RefSeq. We find the match ID, right? And if we look at the match ID, that's going to be this element here. This element here that we find when we click on that link. So as we click on this link, we'll be able to find that match ID. Right? Once we have that match ID, we'll then put the start end and the start and the end. This should go in order. Notice the lower number always goes first. That's the start, and the end goes here, right? So if it's on the negative strand, it's still the lower number that goes first and the end that goes here, but we will just say that it's on the negative strand. So even though on here, you know, this may be the higher number, says start, and this may be the lower number, it says end. We want to make sure that when we put it on the table, we would actually put the end in the first column and the start, because it's a larger number, in the second column. Please keep that in mind. Starts are not the same when we think about them. Same goes here, because this is negative, but this is the lower number, right? This is going to be our start, and this is going to be our end. So it's important that we identify that, and we just make sure that we note that if it is on the negative strand, we're just saying that it is. We'll then put in our e-values here that are identifiable in our searches. DFAM, the e-values listed in the table. On BLAST, any e-value will be listed. If you look at the alignment, it'll be listed right here, right? So this is the expected value, 9 times 10 to the minus 35. So that's what we'll put into this e-value. We should identify what species the thing we matched to was. So all DFAM results will be Danny Rario. For BLAST results, we'll be able to find that information. Typically, it's identifiable here inside. It'll be given here inside of this result. So it'll tell you what species it is. And it'll also be identifiable here on this table. Lastly, we'll put a summary of what that identifier is. In this case, the information you're going to put in, if we look to click on any of these items, right, we would identify this as a TDR25, which is a non-autonomous DNA transposon. So what you would do is you would give, say, TDR225 would go in this position, colon, whatever it is, non-autonomous DNA transposon. So an example would be here, like we have TDR1, and then we have DNA transposon TC1 like element TDR, right? Or we have TC1 DR3 non-autonomous DNA transposon. So that's what goes in this information for repetitive elements. For BLAST elements, we would just copy the name of the target that was identified, RAS association domain containing protein 5, isoform 3, and we would plug it into this last column here. So that's a little walkthrough on how to process and generate these data. We'll talk more about them in class next week, but make sure that you have your tables prepared and ready for processing next week when you come to class. All right, thanks a lot.